All right, well, the attendees number has been pretty consistent, so I think we'll go ahead and kick it off.
All right.
Well, welcome, everyone. Welcome to EcoQuestions. Everyone is currently muted, and this session is being recorded. So if you'd like to remain anonymous, please keep your camera off, which they are, and change your screen name. If you're having technical difficulties, please type in the chat and I'll do my best to address it. I'll be keeping an eye on that. My name is Jenny. I'm the EcoFlora coordinator at the Desert Botanical Garden, and tonight we have our special guest, D. Nathan Taylor. Welcome, Nathan, and thank you for being here. You all may have read about Nathan in the current EcoQuest we have going on, Spot Spurge. He's an avid and very active iNaturalist user with several projects, and as we just were saying, has likely identified a good number of your sandmat observations. Nathan graduated with his master's degree from Sul Ross State University, where he worked in the herbarium with the curator, Dr. A. Michael Powell. Currently a new Ph.D. student at Oklahoma State University, he works as a TA in the Department of Plant Biology, Ecology, and Evolution. His primary interest is sandmat spurges and plants of the high plains of Texas and New Mexico. This evening, Nathan will be sharing his knowledge and enthusiasm about these incredible plants, our local species, identification tips, and then we'll open up for a question and answer session. During the presentation, feel free to type your questions in the Q&A box. This is found in your toolbar, and it looks like two little talk bubbles for an icon, and it's labeled Q&A. As attendees, you can upvote each other's questions, and you can answer each other, and when we get to the Q&A session, we'll answer the top questions for as long as we have time. And with that, I'm going to mute my mic, and I will turn it over to Nathan.
Thank you. Well, today I'm going to be talking about the sandmats, as Jenny mentioned, and that is Euphorbia section Anisophyllum. Now, for those of you who are unfamiliar with this group of plants, you may be wondering what these look like, what these are. And here's a pretty typical example of what you might see in a sandmat when you come across one. You know, it's kind of a weedy thing, and a lot of people consider it a weed, pull it, spray it, you know, try to get rid of it. But there's a kind of intricacy to these plants. This is Euphorbia maculata, the common spotted spurge. And if you look really closely, what you'll find are these little things. Now, these are not flowers, even though they look like flowers, and they are flowering structures, but they're actually inflorescences. So they're a collection of flowers, and it's called a cyathium. Now, this deserves a little bit of a closer look, I think. So let's look at this dissected cyathium. You have a dissected cyathium on the left, two of them actually, and then a whole cyathium on the right. Now, a cyathium is made up of an involucre, which is on the outside. Now, what an involucre is it's just, they're fused leaves, fused modified leaves called bracts. And in this case, there are 5 bracts that sort of surround the flowering structures. And at the tips of those bracts, you have 4 glands. Now, these may produce nectar. Some of them do, some of them don't. And a lot of the times you'll have these petal-like appendages that grow off of the glands. Now, inside of the cyathial wall, the involucre, you have a collection of many staminate flowers. Now, these are the male flowers, and it's also worth noting that these are simple flowers. They don't have any petals or sepals, and they only have 1 stamen per flower. And there can be there can be even up to 100 of these things in a single cyathium, but usually is at least 5. And each of these have their own little pedicel, their own little flowering stalk. And then in the center, you have a single pistillate flower, a single female flower that produces the fruit and the seeds. And this comes out of the center of the cyathium and usually sort of curves over into the area that lacks a gland. So if you look over here, there are the 4 glands, and then there's this missing gland here. This is called the rudimentary, quote, fifth gland. But it curves through there, and it stands upon its own pedestal. And there can be many of these flowers per cyathien. Now, there's a lot of diversity of cyathia in Euphorbia and in section Anisophyllum. And a lot of the times you'll have these, I mentioned petal-like appendages before, and if you look at this one right here, you can have really large ones, you can have pretty much non-existent ones in the petaloid appendages, like the one in the lower right-hand corner. Sometimes the appendages can be sort of deeply incised, or sometimes they can be entire. Sometimes the cyathia are densely clustered, sometimes they're solitary. There's a lot of diversity here. Now, cyathia are basically what makes Euphorbia Euphorbia. That is sort of the single defining characteristic of the genus Euphorbia, the broader genus Euphorbia, the spurges. Now, in section Anisophyllum, as I mentioned before, there are 4 glands, and that is with maybe 1 or 2 exceptions, all of the species have 4 glands. Now, that's not the only thing that makes section Anisophyllum different from other euphorbias. So there are actually a lot of characteristics that make them different. The first one I'll go over is this thing called sympodial branching. And that's kind of a technical term, I understand that, but it's not that complicated. So if you think of let's call it a typical stem, what you have is just a straight stem that grows up and it produces, branches sort of as you go up. That's not what happens in sympodial branching. Basically, that main stem, it grows, produces a single pair of leaves in this case, and then it doesn't grow any further. And instead it has to grow lateral branches off the sides of the tip of that branch. So what you end up with is sort of a series of forks of branches is what it looks like. Now there's some species that sort of get around this by, only producing or only making one of these branches dominant instead of both of them. But this is a common characteristic of the section. And this even happens in the seedlings. So in the seedling, you have a second apical meristem, that's the main stem that the plant grows up with, that's completely absent. So basically all of the upper growth occurs from these lateral branches that come off of the seed leaves. So it's a really weird condition. And this leads to an interesting phenomenon where the branches are dorsiventral. And basically all that means is there's a clear upper side and a clear lower side to the stem. And this is exaggerated by the asymmetric leaves. So in section Anisophyllum, the dorsal side of the leaf is actually longer than the ventral side. And so that basically helps the plant capture more sun. And this is fairly unique, except for some other species in subgenus Esula, another group in Euphorbia. But in those cases, the longer side of the leaf is actually on the other side of the leaf, so the lower side of the stem. Now, another thing that's unique about section Anisophyllum is that it has opposite leaves. So basically the leaves are just held in pairs. And this is usually pretty rare in Euphorbia, except for this section and except for in inflorescences. Now between each pair of leaves you have these little stipules. And these are non-glandular and they don't create spines. They're not prickly, they're not stiff and prickly. So another thing about the leaves is they have this interesting maze-like pattern that you can see sometimes, especially in plants that are not getting enough sun, and that is called Cron's anatomy. Now, what is that? basically all it is it indicates that there is C4 photosynthesis. And how it indicates that is the darker regions are the bundle sheath cells, and that's where photosynthesis takes place. All the lighter areas in between, that's where the CO2 actually enters the cell, because in C4 photosynthesis, it's a process that separates the CO2 capture from the actual turning of that CO2 into sugar. And it's a useful characteristic for saving water and being more efficient. So now with leaves out of the way, let's go on to seeds. So all species in this section, with a few exceptions, have four-sided seeds in cross-section, and they lack a caruncle. Now, what is a caruncle? A caruncle is basically an outgrowth of the hilum. Now, what is a hilum? I get that this is getting pretty technical, but think of it as a belly button for the seed. It's basically where the seed meets the vascular tissue of the fruit. And so if you were a seed and you had an outgrowth of your belly button, then you would have a caruncle. So, so with that, let's look at the final characteristic of the seed, and that is seed mucilage. So basically, these seeds are sticky, and they produce this mucilaginous coat that allows them to stick to bird feed or sand or anything they're growing in. And with that in mind, sort of the idea that it can stick to bird feed and that might aid in dispersal, you might want to know what kind of ecological studies have been done on this group. Well, it turns out not a whole lot. There have been a few examples of researchers studying the dispersal of seeds by ants, and maybe a few on the allelopathy of the plants, but not a whole lot. The one really good exception to this was a pollination study done in the 70s that was actually done in Arizona. So this is particularly relevant. And it's really the only big pollination study that I think has been done in this section. But what they found is that there are at least 10 oligolectic bee species. And oligolectic means that it specializes on the pollen of these, on collecting the pollen of these species. And one of my favorites in this group is this little bee called Perdita minima. And that's what's figured on the right here. And this is actually the smallest bee in North America and comes close to being the smallest in the world, but there are a couple of species that beat it out in Australia. So, sorry. Now that you know what characteristics make up section Anisophyllum, and a little bit about what make them interesting, let's move on to identification, because that's where the real fun begins. So there are several things that you need to take into account when you're trying to identify these plants. And I wanted to go through every single species that occurs in Phoenix, but there are too many species for us to have time for that. So I'll go over some of the basics with you tonight. So first thing to look out for are the leaves. The main thing that you can ask yourself first is, are the leaves entire? Basically, do they lack serrations or do they have serrations? If they have serrations on the leaves, then that falls into one group of species, and if they don't, that falls into another group of species. The other thing is if they're linear or if they're broader. So in the case of these linear leaves, there are only about 2 species in and around Phoenix that have these linear leaves, so that narrows it down pretty easily. The second thing you need to look out for, though, are the stipules. Now, the stipules come in many different shapes and sizes. And in this case, it can be fused into this sort of glabrous scale-like appendage, like in Euphorbia alba marginata. Fun fact about that species, it's one of only 2 species that root at the nodes. That's also known as rattlesnake weed. But back to the appendages, they can also be distinct, like they are here, as in some of the stipules of Euphorbia hypericifolia. And in some cases, they can be divided on the upper side, but united on the underside. So if you're ever trying to key out these species, it's important to look at the upper sides of the stems instead of the undersides. Or if you do look at the undersides, just know that if they're distinct on the upper sides of the stem, to go with the couplet that says that the stipules are distinct from one another. Otherwise, that'll make things a lot more complicated, and you're going to have to do a lot more research to try and figure out what that species is. So the other thing, or the final thing that you really want to be aware of are the hairs. Now, if you get nothing else out of the identification characteristics slide, pay attention here, because this is where things get really interesting. The hairs can be either straight, or they can be oppressed, or they can even be glandular. And even more than the type of hair, the placement of the hair is really important too. So there's some species that only have hairs on the stems. There's some species that have hairs on the stems, leaves, and fruits. And there's some species that don't have hairs at all. So in this case, you have Euphorbia micromira that doesn't have any hairs at all. You have Euphorbia arizonica that has these sort of glandular hairs. There are only 2 species that have glandular hairs, by the way. You have this species, this is Euphorbia pediculifera, that has these sort of grayish hairs, these dense grayish hairs. And then you have Euphorbia prostrata that just has these straight hairs that are coming off of the surface of the fruit. So with that out of the way, I also want to mention habit. There are a couple of characteristics that a lot of people use and think are really important, but really have to be used with some caution. And one of those things is habit. So you can have these clear prostrate plants, these plants that, you know, are very, very flat on the ground. And then you can have species that are more upright like this. And that is a useful characteristic for identification. But it's worth noting that any plant that is prostrate, with the exception of 2 species, can be upright in shade. And any species that is upright can appear more prostrate if it's been trampled, and it has to regrow after being trampled. So you really have to be careful about using that characteristic. There is another characteristic that you have to be careful with, but can be useful, and that is the maculation. So basically, some species will have this spot on the leaves, and that can be useful for telling what species it isn't, but it isn't necessarily easy to tell what species it is by it. Now, what do I mean by that? Basically, species that can produce maculations may not produce maculations. They either they can produce them, or sometimes they don't. It just depends on how the plants are growing. But there are species that can't produce maculations. So you can only use maculations of a character as a characteristic if they are present. Otherwise, you're going to end up thinking it's something that it's not. And just so you know, anything that has a spot on the leaves is not spotted spurge. Some people will go with that, so just remember that. Okay, now let's look at a couple of examples. So in this example, you have 2 species that look fairly similar, I would say. Now there are some things that you can see that are obviously different, like for instance the maculations here are a little different. There's some sort of light gray lines on the leaves that you can't see here, but otherwise they look pretty similar. Now, how would you go about telling these two apart? Well, before I go to that, I just also want to note that there are even some hairs on the stems here and some hairs on the stems here. But if you really want to know what these are, you got to look at the fruits. So on the plant on the left, it doesn't have any hairs on the fruits. On the plant on the right, hairs on the fruits. And that can lead you to understanding that the plant on the left is Euphorbia abramsiana, and the plant on the right is Euphorbia maculata. Remember those hair characteristics that I mentioned. They're super important for telling these species apart, and that's how you tell the species apart in this case. Now, let's look at another example. So you have two upright plants that both have no hairs on the fruits, they have no hairs on the stems. Maybe every once in a while you'll see a hair or two on the plant on the right. How do you go about telling these two apart? I mean, from what you can see, there's maybe a few differences. Like here, you have Cyathea that are densely clustered on the left, and they're a little less densely clustered on the right. But sometimes that can lead you astray depending on the condition of the plant. So how would you go about really telling that these two species are different? Well, for that, you really want to look at the stipules. So if you look on the left, notice that the stipules are spreading. Now, they also happen to be fused in this example. They don't always have to be, but they're spreading. They're spreading outwards away from the stem. And on the right, they're pretty closely oppressed to the stem. So based on this information, you can actually tell that the one on the left is graceful spurge, Euphorbia hypericifolia, and I think it's hyssop leaf spurge, Euphorbia hyssopophilia. It's also worth noting that the stipule on the left is a lot longer than the stipules on the right. So again, look for stipules and hairs. Those two will get you pretty far when trying to identify these things. Now, I also want to go over three species specifically that I think might cause some trouble if you're just looking at the pictures on iNaturalist. So the first example is Euphorbia polycarpa. This is small-seeded sandmat. And the reason I picked this is because the profile pic for this species is of a variety that occurs in southwestern California. And so it has these long, showy appendages that look like Euphorbia albimarginata, rattlesnake weed. But the Euphorbia polycarpa in Tucson, Phoenix, Arizona in general, I guess, they have these much, much shorter appendages, more like that. So just be aware of that if you're looking at some of the pictures on iNaturalist. So the second species that I want to go over is Euphorbia revoluta. The reason I want to mention this species is because it's not actually mentioned there's not actually an observation rather of this species for the Metro Phoenix area yet. But it has been known to occur in the area after looking at some SciNet observations.
I just want to say that that's a new goal for everyone listening, to go out there and find that. And also this photo was by Steve Jones. Yay!
Yeah. This one was by Steve Jones, and he's really a really good resource for the region. I'm sure if you haven't come across him on iNaturalist, if you post observations, you probably will. Yeah, he's really helpful. Now for revoluta, the main way you tell this apart from everything else is the linear leaves. This is one of only two species that has it. The other species has serrated linear leaves, and that is Euphorbia florida. Also, the other thing to keep an eye out for is the Cyathea. Now, the Cyathium, they don't really have very showy appendages, whereas Euphorbia florida does. It has it's one of the showier species in Arizona. So this is a good thing to keep an eye out for. This typically likes to occur in more mountainous regions, I think. Really rocky soil and mountainous areas. So there's one more species that I want to mention, and actually this is not so much for identification purposes, but because it is something that you can keep an eye out for that is not very common in Arizona yet. So this is Euphorbia ophthalmica. I think this is Florida hammock spurge, I think is the common name. And the reason I bring up this is because this is a very recent introduction, or a relatively recent introduction to Arizona. And there are only, I think, maybe 2 or 3 observations in Phoenix so far. So any observations of this would really kind of help document its spread in Arizona. This is typically spread through nursery plants, and in some places it can become established outside of cultivation, or outside of cultivated potted plants. In some places it can't. So this is a good species to keep your eye out for. More than likely you'll only find it in sort of well-watered areas that have been recently have had recently been planted with landscape plants. Let me put it that way. So yeah, the way you tell this apart from others is that it has these densely clustered inflorescences, and they sort of terminate the branches. They're not sort of held along the stem, but they really just, they produce the inflorescences right at the tip of the branches. There is one other species that sometimes does that. It's a native, and it's Euphorbia capitulata, but it usually has long petaloid appendages, long petal-like appendages. So keep an eye out for that. All right, and I think, yeah, so that's the last slide. So with that, some final thoughts. I hope that looking over these slides and looking at the structure of Euphorbia section Anisophyllum has really gotten you more interested in this and given you some appreciation for the group. And at the very least, made you not hate it quite so much if it's in your lawn or garden. So with that, I'd like to thank Oklahoma State University and the Fishbein Lab for giving me some comments on this presentation, and thank you to EcoQuest and Jenny Davis for inviting me to present here. Thank you.
Thank you, Nathan. That was fantastic. We've got 1 or 2 questions. I have one that came in with a registration, and you may have kind of gone through this in the presentation, but in the In Defense of Plants podcast that you were on, which was also fantastic, you mentioned that leaves on a plant are variable and develop a pattern of sets and leaves. And the question is, please talk about what constitutes patterns and sets to help me be sure my photos include enough material to be useful.
Okay, so that's a really, really good question and a complicated one. So basically, let me see what's the simplest way I can put this. So let's start out with a plant that develops from a seed. So it produces the seed leaves, and then it produces its first pair of true leaves, and then it branches out and produces the first sets of leaves. Now these leaves are often broader than the leaves that occur once the flowers start. So, or once the cyathea are produced. The easiest way to think about this is sort of pre-cyatheal and post-cyatheal leaves. But that's not entirely true. There's sort of a gradation between the two sets of leaves, but that is the simplest way to break it down, is pre-cyatheal and post-cyatheal. It's also worth noting that in perennial plants, a lot of the times the the leaves that I would think of as pre-cyatheal are kind of absent. And so you just have to go on based on other characters for that. Now, I will mention that luckily in Arizona, the species are so distinctive that you generally don't have to mess with that.
That's good to know.
Usually you really only have to worry about the different sets of leaves in places like, or in really difficult species complexes. Especially in Mexico, there are a lot of really weird species complexes that that make you want to kind of look at that. I hope that answers the question.
Sounds good to me. We had a question in the chat before the Q&A started coming in. How many species are there in Arizona?
Oh man, I think there are like 30 something, if I remember right. It's been a while since I looked up the number, but for some reason the number 37 is coming to mind.
Wow. Any guess at how many of those are native?
Actually most of them. So Euphorbia maculata is introduced almost certainly. Euphorbia serpens is probably introduced. Euphorbia ophthalmica is definitely introduced. But I think most of the rest are actually native.
Cool.
So really high native diversity.
I think a lot of people are very surprised by that. I think they see
Yeah.
all the weeds that are invasive.
Uh-huh. Yeah, you know, there are a lot of native species in this group. Yeah. Yeah, and I mean, Hipperocephalia, I guess, is another one, but that's really like only 4 out of 37. I mean, that's, you know.
That's pretty good odds.
Yeah, yeah.
Next question is, can you share what role spurge plays in our environment? I know that might be kind of tough because we don't have that data, which is
Exactly.
really kind of what we want to know.
Right, and, you know, there really isn't that much information. Now, if you want to be somewhat speculative about it, you could say that they do hold down the soil.
Mm.
Now, the one thing about that is some species are allelopathic. But unfortunately, only 2 or 3 species have been studied for allelopathy, and those were the invasive, the introduced species.
Which I guess is important to know.
Yeah, so
First.
it's not really clear whether that would like inhibit other ground covers. So you know, you could say that it holds down the soil, but you could also maybe make the argument, if more data comes along, that it also keeps you know, new species from holding down the soil. Now, one thing I will say, there's a lot of evidence that it's an early colonizer to sites. They just, I mean, you can see this in your lawns and gardens. I mean, they grow really well in disturbed soils, and they do well in arroyos as well. So
if you have this as a ground cover, it might be your only ground cover.
Possibly, depending on the species, and you know, depending on if all the species are allelopathic or just one.
I don't know. Sorry, I don't know if we mentioned what allelopathic means. That is basically, correct me if I'm wrong, chemicals that exude from the plant that keep other plants from growing around it.
Yes, exactly. Basically, it's plants producing herbicides.
Yeah, that's a perfect way to put it.
Yeah, yeah. And in euphorbias, basically they actually excrete the allelopathic chemicals from the above ground part of the plant, which is kind of strange.
Yeah.
But when it rains, that seeps into the soil and suppresses seedling growth. So that's why you don't have a lot of species germinating under sand mats.
Right. Which also makes them kind of amazing. Like I said in the EcoQuest, evolutionary champions. They have so many things that they've managed to come up with. Steve Jones commented 29 species in Arizona. Not counting the one you just mentioned. That's a recent introduction in the ASU herbarium.
Okay. I was probably thinking of another state for 37 then. That's a
still
good comment.
impressive.
I will also mention that there is another species, Euphorbia villifera, that I've recently discovered for Arizona. Actually, two more that I've recently discovered for Arizona. So that would bring it up to about 33. So yeah, I think Rayturneri and both Euphorbia rayturneri and Euphorbia villifera now occur in Arizona.
I think touching on that question still of, to that, smallest bee in North America, this plant probably matters a whole lot. So a lot of times we have, we look at things very anthropocentrically and what does it benefit us? But, they do play a role in the ecosystem, whether it benefits us as humans or not.
Mm-hmm.
So next question is, what is the function of the syatheal gland?
So it kind of depends. Like I said, in some species they produce nectar, in some species they don't. And in some species they're so small that they don't really seem to have that much function at all. Now, to be clear, any real indication I'm giving as to what the syathia or what the glands are used for, at least in section Anisophyllum, is speculative, because again, no one's really studied it. But they do, it would, because of how they look, like for instance here in Euphorbia capitulata, they're sort of dark red. There's some other species that have those dark red glands as well. You can imagine that those are useful for attracting pollinators.
Yeah.
And of course, that's where the petal-like appendages come out of, which are, they're basically simulating petals. So they most likely have a role in attracting insects.
I think this is a great call for community scientists to get on this stuff.
Mm-hmm.
Steve Jones said maybe you were counting all taxa Nathan 34 in ASU, not counting the 3 new guys, so you were right. Steve, you're so fast.
Yeah, I'm surprised I ended up so precisely accurate. I figured I would be off by at least one.
Next question is, what time of the year do we normally see spurge in the Sonoran Desert?
Right. So it depends on the species. For the perennials, you can see them, I think, most of the times of the year when it's not, most of the years during the growing season. I've seen some observations that have been taken during this month that look like they've been hit by frost, but they're still doing okay. So some species can, actually keep growing a little bit after frost, but generally it's during the growing season for the perennials. Now for the annuals, most of them seem to germinate during the summer and grow and are most easily visible during the fall, early fall. August is generally what I consider the Anisophyllum month.
That's easy to remember, 2 A's, August Anisophyllum.
Yep.
I know, I saw one, I think I put it on iNaturalist, but it was like a purplish color and it looked half dead. And I thought that was really cool. I thought it was a really neat color change for that. I don't know what happened. I imagine possibly drought stress after this summer. It's been so bright.
Right. Yeah. So basically, when the plants turn purple, basically that's an indication either of cold stress or sometimes drought stress or sometimes too much sun exposure. That's basically the production of anthocyanins, and those can serve as sunblock or some other.
That's our fall color in the desert.
Mm-hmm.
Next question is, how have you used both herbarium specimens and citizen science data in your research so far?
So that's a good question. Now, as far as like the raw data itself, I haven't actually used a lot as in terms of numbers. Now, what I have used them for is getting a better understanding of the structure of the morphology of the species. One thing I really like to do is try and figure out how to identify these plants even under the worst circumstances.
Mm.
And what citizen science has done, particularly the observations on iNaturalist, is they've just broadened the pool of available data points to understand the morphology so much. I mean, I can go online and look at 1,000 Euphorbia maculata observations if I want to, and it'll actually several thousand at this point. And you know, that means that there is the possibility to actually identify these things under almost every set of conditions you could think of. And so what that allows you to do is to understand what environmental conditions sort of play a role in affecting these morphological characteristics that you're determining your IDs on. And actually, I have a really good example of that. Let me scroll down to Euphorbia prostrata. So in this species, you'll notice that in some individuals, the hairs are actually oppressed on the fruits. And this is a key characteristic in taxonomic keys, is that the hairs are straight instead of oppressed. And there's a mess. So yeah, they're straight instead of oppressed. But every once in a while, there are conditions that will lead to the hairs being oppressed like this. And that's something new. That's something that hasn't really been noted before.
That, I guess, never really, we think about citizen science a lot of times in terms of data, and numbers, and, just being able to have access to these images and like crowdsourcing these images is super important.
Well, and another thing I'll mention is that even though I haven't used the data yet, one thing that I can certainly do is just take a bunch of the data that's here and do a niche modeling analysis on it. So basically just sort of get a better idea of where these plants occur, what conditions these plants occur under, and then compare them to each other. And that's something I'm actually thinking of doing during my time here at OSU for some species, not all of them. But
that would be really interesting. Exciting. Next question from Steve. Are you aware of any galling critters on these species?
Galling critters. Well, let's see. I would ask you if you could be more specific. Let me think here. So there are whiteflies. Whiteflies will sometimes make their home on these things, and sometimes you can actually find their pupa, and their pupa look really cool. They look prehistoric, but and you can also find the parasitic wasps that eat the whitefly larvae. You can find, of course, aphids and ants. Ants love these things. They'll spread the seeds. They'll, I guess, take the seeds away.
Which I think is an important statement to the ecological value, for lack of a better term, is, I mean, so much that ants do for us that we don't even realize.
Right. Yeah, and I mean, ants, I don't think a lot of work has really been done on, what all species, but it's actually even been suggested that the seed morphology has sort of evolved to fit the preferences of certain ants.
Wow.
To make them look like beetle larvae, and certain types of beetle larvae to make them more attractive to being spread by ants. Now,
That's
of
so
course,
cool.
that hasn't been studied. It's just been speculated, but it's something interesting to think about.
Very. Sam says some rusts, fungal rusts. Yes.
Mm-hmm. Yeah, there's one particular rust that what it does is it makes the stems stand straight up. Even the species that won't stand straight up in shade will stand straight up when this fungus infects them.
Wow.
Yeah, the plant stem goes straight up, and that allows, I guess, more airflow for the spores to be spread.
That's really cool. I
Yeah,
just, in my head, envisioned, Looney Tunes when something goes over all of a sudden.
mm-hmm. But no, it's a really weird rust, I should say. I think the common name on iNaturalist is Chamaesyce rust, if you want to look it up.
Makes sense.
So, yeah.
Also reminds me of the mind-altering fungi that tell ants to go climb up on a tree and sit on that branch, and
Mm-hmm.
the zombie ants.
Yeah.
Next question, is there anything that eats these spurges?
Yeah, so mule deer have been reported to eat a few of the species. At least I think it's Euphorbia prostrata. I think it's been documented to eat this species. Not many species though. Yeah, there really aren't a lot of documented cases for for animals eating these things, except for insects. So that's another really good opportunity for citizen scientists to sort of see what's going on with these things.
I'm shocked that anything does with that, latex milky sap. That's
Mm-hmm.
like the reason for it, is to keep things away.
Right. Actually, one other thing that comes to mind, I think there was an observation of some quail species eating it in California. I don't remember which one. And I don't remember which species either.
Tom Gatz says desert tortoise. And Steve said he's seen sphinx moth caterpillars nibbling at them as well.
That's right. There have been, Hyles lineata does occasionally use this plant. It doesn't seem to like it. It's not its first preference, and Hyles lineata will eat just about everything. But it loves 4 o'clock, not 4 o'clocks, evening primroses.
Yeah.
But yeah, that's the common hummingbird moth.
Next and final question that we have, last call for questions, anyone? We're a little bit over. How deeply do the roots grow?
So that depends on the species, but most of them only grow about, I would say, a max of certainly no more than a foot. Usually more like, I don't know, 8 inches or so. Of course, I haven't dug up many of the perennials that deeply, but certainly the annuals don't get very deep.
All right. I think that's all the questions that we had. Thank you so much, Nathan, for joining us tonight and graciously sharing your expertise and knowledge and experience. And personally, I've learned a lot. And I think everyone here can say the same.
Well, thank you for having me. This has been a lot of fun.
Thank you, everyone, for attending. This is recorded, so everyone that's registered will get a recording of this presentation. So keep an eye on your email. And thank you again, and have a great night. A lot of thank you comments in the chat box. Thank you for your time. I appreciate these little guys so much more. Goal achieved.
Great.
Thank you. Very helpful, even in Florida. Thank you, Nathan. Great presentation.
Thanks, everyone.
Thank you so much. Have a great night.
You too.
Bye, everyone.